The overall goal of this study is to examine thalamocortical connectivity in albinism and controls using diffusion imaging and to compare the output optic radiation reconstruction of two tracking algorithms.
Diffusion MRI and tractography can help answer key questions in vision research, such as the effect of axonal misrouting on structural organization of the visual pathway in human albinism.
The main advantage of this technique is that it allows non-invasive mapping of large white matter pathways in the living brain and has shown promising advances in neurosurgical planning.
Acquire all imaging as described in this protocol on a 3 Tesla MRI scanner equipped with a 32-channel head coil. Prior to imaging, first thoroughly screen every participant for MRI safety and have them sign a consent form describing the protocol. Provide the subjects with earplugs for hearing protection before positioning them supine and head first on the scanner table. Provide an alert squeeze bulb. Then place cushions to reduce head motion. Landmark above the eyes at eyebrow level before sending the subject into the scanner. Begin imaging by acquiring a high-resolution T1 weighted image using a 3D MP-RAGE sequence covering the entire brain. Use the parameters seen on screen here.